most people have pretty much worked with you as well, so they know what you're about.
Yeah. And this is focusing just on that, not on RNA-seq and genome assemblies and nothing like that, so just one area. Yeah.
That's good.
I just asked Gary, do I need to turn it on or is it?
It's already on. Yeah.
Oh, right. Gosh, so whatever I'm saying now is already on, getting streamed. Uh-oh.
So be good.
Okay, good afternoon, everyone. Welcome to today's Rothamsted seminar. Today we have K1 talking to us. He is the head of bioinformatics and founder of Netminer. He has a PhD in bioinformatics and genome research from Rothamsted University and has worked in bioinformatics and software development for 10 years. Today he'll be presenting his work on an intelligent data model known as the Netminer knowledge graph.
Okay. Right, test, that's working. Okay, so hello everybody, hello everyone at Harpenden, Norswijk, people watching on YouTube. My mom is streamed in, it's the first time she'll be listening actually, what her son is saying. So it's nice to have you here. So one thing that was not mentioned is today is my 11-year anniversary at Rothamsted, so exactly 11 years ago. I joined the BAB department at that time, Bioinformatics and Biomathematics it was called. So I've been working with many of you in the past, my team has been working with many of you in the past, and today my talk is about one area of research we are doing in my team. So just give you an introduction and some background. Researchers at Rothamsted and around the world are working to push the boundaries of human knowledge and tackling key challenges like food security, agriculture, and climate change. So you would think they have access to the best available tools to help them on their quest for knowledge. But in reality, this is not always the case. The research tools that we've got in our hands, they are often just substandard and make it just actually very tedious to find information. So in this seminar, I'm going to present how we developed an intelligent data model, which we call the Netminer knowledge graph, that will help you as a researcher to find information quickly and more easily. Okay, so I've got a technical challenge here. I'm connected to two systems. One is this, one is my laptop. So my talk is structured in 5 sections. I will first talk about biological knowledge discovery. What do we understand with biological knowledge discovery? Then talk about what are graphs and how are they useful. Then explain and introduce the Netminer knowledge graph. And finally show some use cases and discuss future work. That's working, great. Okay, so biological knowledge discovery. There are many types of biological knowledge discovery. For example, drug discovery, trait discovery, gene discovery. Today, I will be mostly talking about gene discovery, but it's applicable to other areas of research discovery as well. So what is gene discovery? Let me give you a metaphor. So genes are rarely single actors. To explain the metaphor, unraveling the roles of genes in complex trait genomics is often similar to unmasking the heroes and villains in a plant biology whodunit. So multiple genes are generally involved and play different roles. How they interact reveals the plot lines in the trait story. So what is it we are doing? We are building software and data resources to help automate the process of explaining the plot lines and unraveling the story behind complex traits and trait biology. So we are creating computational tools that bring different types of evidence together and then weigh up competing storylines to present the user with the most compelling stories. Okay, so this job of assembling evidence is a tedious job for humans. Machines can help, but they face their own challenges. So let's review for a moment the type of evidences needed to go from data to understanding. So some of you might know Clifford Stoll, an American astronomer. He was saying, data is not information, information is not knowledge, and knowledge is not understanding. So what does he mean with that? So let's talk about data. So data comes in many different formats. Oh, sorry. You need to tell me when I'm switched the slides on here, but not on there, okay? It's got two systems. Okay, does that make more sense now? So I'm talking about the differences between data, information, knowledge, and understanding, okay? So there are different differences. Say data, for example, comes in many formats. We've got Excel spreadsheets, FASTA, GFF files, tabular, XML, you name it. Data is not always fair. It's not easily findable, accessible, interoperable, reusable. So that's a big challenge. Information is in many locations. It's highly interconnected. Over 1,600 biological databases have been published in a recent issue of NAR. So it makes it hard to find and interconnect information. Knowledge itself is when it gets actually published. It's a bit more understandable. So knowledge can be buried in free text, like the scientific literature, or it can be structured in pathways or ontologies, which are computer machine readable. So there are over 29 million publications in PubMed, and there are over 750 different ontologies published on BioPortal, for example. So this data, information, knowledge all together then can be used to derive understanding, for example, to understand yields, nitrogen use efficiency, and disease resistance. We need all these components to come together to understand our traits. Okay, so what we've been working on in the last years is a software tool called NetMiner. It stands for Knowledge Network Miner. The K is silent. And it's a web app that makes biological knowledge discovery a bit faster and a bit more fun. So it is used by researchers at Rothamsted and around the world. 68% of our users are coming outside of the UK. So myself, I'm leading the project. Chris is a co-lead. Ajit is responsible for the front-end development of NetMiner, and Marco Brandisi works on the back-end technologies and interoperability of NetMiner. Okay. Right, so if you haven't seen NetMiner before, this is how it looks like. It's a simple search interface, and you can search it for specific keywords. It's like a bit like Google. You can search it for gene lists, or you can search it with genomic regions or combinations of those three. I will now play a very short video that just gives you an idea of how the search looks like. So you can search for seed germination, seed dormancy. It gives you a ranked list of candidate genes. And if you click on a gene, it presents you the knowledge about that gene that you can then interactively explore and find relationships between genes, traits, diseases, and many other types of information. Okay, so these are publications. Right, so this is just in a loop. So what NetMiner really does, the key bits are it ranks information, and it visually shows knowledge maps that are comprehensive and are coherent. So all information is one single view. So the rest of my talk is really talking about how NetMiner actually works. So NetMiner is not a black box. It's not like we put a lot of data in and it magically tells you, try this or that. So it is powered by a large integrated knowledge graph and graph analytics to search and visualize knowledge. Right, say, okay, that's there, cool. So let's do this. Okay, so let's talk a little bit about graph analytics now. And I'll try to give you an introduction, which is gives you the idea of what graphs are without going into too much technical detail here. So graph analytics has a history dating back to 1736, when Leonhard Euler solved the 7 bridges of Königsberg problem. Okay, so what's the problem? The problem asked whether it was possible to visit all 4 areas of a city connected by 7 bridges while only crossing each bridge once. Okay, so at the top, you see Königsberg. These are the 4 areas, and green are the 7 bridges. So with the insight that only the connections themselves were relevant, Euler set the groundwork of graph theory and its mathematics. So he had this idea to model this problem as a network. So what you see on the right-hand side is representing Königsberg, and the 4 areas are nodes, and the 7 bridges are represented by edges. Okay, so very simple, but a very clever idea to model this as a graph, as a network. So Euler modeled the problem as a graph, and then he showed mathematically that that there is not such a walk where you can cross all the 7 bridges. How did he do that? So he proved that in any graphs, such walks are only possible if exactly 0 or 2 nodes have an odd number of edges. Okay, and if you look at the map of Königsberg, you'll see that all 4 nodes, they have an odd number of edges. So this node has 3 edges, this node has 3 edges, one has 5, and the other one also has 3 edges. So because of that, he could show actually that it's not possible to have a walk through the town. So, but graph analytics did not, so it was 1736, but graph analytics did not catch immediately. It was 200 years until the first graph textbook was published in 1936. Then in the late 1960s and 70s, network science and applied graph analytics really began to emerge. So in the last few years, there has been an explosion in interest in graph technologies. A recent survey indicated that 69% of companies are planning to implement graph databases in the next 12 months. So the demand is definitely accelerating based on the need to better understand real-world problems. Thank you. Right. So this was 1736. Graphs looked, and Euler worked on a very simple graph. Since then, now, we're working on a different scale of networks and graphs. So networks are a representation, a tool to understand complex systems and the connections inherent in today's data. So the top here, this is showing you a simple graph of proteins and their interactions. And over here, you see a social network. So you've got nodes are people and relationships are their friendships. So you can represent how protein or social systems work by thinking about interactions between pairs of proteins or pairs of people. By analyzing now the structures of a graph, we can answer questions and make predictions about how the systems work and how individuals behave within those systems. So in this sense, network science is a set of technical tools applicable to nearly any domain. All right. Okay, so graphs are one thing, and then graph algorithms are the other bit. So most graph algorithms can be classified in 3 major groups. There are path-finding algorithms, centrality algorithms, and community detection algorithms. So in NetMiner, we use all 3 types of algorithms. For example, we use path finding to find the shortest path or evaluate route availability and quality. We use centrality to determine the importance of distinct nodes in the network. And we use community detection to lay out our networks and visualize the data. So the very special type of graph that I'm focusing on today, it's called the knowledge graph. And I'm going now to spend a minute just to introduce what a knowledge graph is. So this is a very simple knowledge graph with 3 nodes. One node is Rothamsted Research. The other 2 nodes are Harpenden and Norswick. So each node has a label. So Rothamsted Research is an institute, Harpenden and Norswick are sites, and the relationships between them have a label as well. So Rothamsted Research is based in Harpenden, and Harpenden is about 230 miles away from Norswick. Okay, so this is really a knowledge graph example. There are different ways of how we can generate and model information in a knowledge graph. For example, this information here that Rothamsted Research was established in 1843 can be a property of the node Rothamsted Research, or we can say we model it as a separate node as well. So we create a relationship called it founded in 1843. Sorry. Let's do this. Okay, founded in 1843 and represented this way. So it's really hard to model information and knowledge in a coherent way, and it's always good if you're planning to build a knowledge graph to work with a knowledge engineer to advise you on how to model and represent information. Okay, so now coming to explain a bit, so what do we mean with things but not strings? So if you think about it, if you search for a term Leonardo da Vinci, it is in first place just a string. But once you've got a knowledge graph and you identify Leonardo da Vinci in the knowledge graph as a node, it then becomes an entity, a recognized entity. So we can now go and find everything that is related to Leonardo da Vinci. And next step would be we can go and ask the question, what is the nature of the relationship between Leonardo da Vinci and things that are connected to it? So in knowledge graphs, we've got proper things that mean something to machines and computers. They are not just text that is very hard for computers to understand. Okay, so Google, for example, they're building their own knowledge graph. And let's take a look how the knowledge graph, Google's knowledge graph, for example, can be used. So if you go to Google and search for Matt Groening, you might actually make, next slide. You might make some unexpected discoveries. You might learn a new fact or new connections that prompt suddenly a whole new line of inquiry. So, for example, did you know that Matt Groening, he is the creator of Simpsons, younger generation might know. So where did he get the idea for Homer, Marge, and Lisa's names? If you look at the knowledge graph, it's a bit of a surprise. You can see that his parents were called Margaret and Homer. His sibling was called Lisa. So it gives you that idea, okay? So the intelligence of the knowledge graph is really between the relations between the entities. That's the key. Right, so that was my introduction into graphs, very short introduction into graphs and graph analytics. Now I'm going to focus a bit more on the Netminer knowledge graph, what information we capture and how we build the knowledge graphs in Netminer. Okay. All right, so the orange circles are the types of information we currently capture in Netminer. So we are mostly focused on genes, RNA, proteins, SNPs, metabolites, publications, phenotypes, and their functions. The green bits are species that we are also capturing partially in Netminer in our knowledge graph. There are lots of other bits of bits of information that we currently actually don't capture, but we might do in future, like fertilizer, pesticides, germplasms, varieties, and then all this area, like environmental data, soil, weather, time, trial, field management, is currently not captured in our knowledge graph as well, but might be in the future. So the first thing is, when we want to build knowledge graphs, is to decide what sort of information we want to capture, and then think about where can we get the relationships between the different entities. So, for example, in our wheat knowledge graph, this is showing you an overview of where the relationships are coming from. Say, different databases have different types of information. Say we for example, get some relationships from ensemble plants. Some information is coming from pre-published data that is part of designing future wheat. Other bits of information are extracted from text, from publications. And then Netminer itself has some algorithms to create intelligent relationships between those entities. So it still might be a bit abstract. That's why I'm going a bit more detail to explain how we go from data now into a graph representation. So let me explain the columns. So these are the columns of a spreadsheet. It's a genome-wide association study data set. So the types of information we've got in the spreadsheet are SNPs. Then we've got p-values, alleles, distance to transcript, chromosome name, position, gene, consequences, and the phenotypic description. So we've got about 66,000 SNPs, 20,000 genes, and 107 phenotypes. The relationships between the columns here are hard to understand for machines. So a machine is giving this file, it doesn't really easily can figure out how the different entities relate to each other. If we now go and transform this table into a graph representation, into a knowledge graph, then the relationships between the different entities, they become explicit in a database, and a machine can now much easier understand what they mean. Say, we've got now a gene, we've got the semantics saying it's close to a SNP, and the SNP is associated with a specific trait. So it helps machines to understand the data better, but also to users, it helps them to see connections and relationships much easier. So if we go and add now a bit more information to the graph, then you can see that two genes are connected because they're linked to the same trait. So now we can go and extend this small graph with another data set. We can go and bring in some protein-protein interaction data from BioGrid and overlay it onto the pre-existing knowledge graph. And what you can then see is that things suddenly get connected, and the biological story that explains maybe storage 56 is a trait, explains the story in a more coherent way. Say something that we might not have thought about it might become more obvious. Okay, so this is just giving you an idea of how the networks are generated that are in Netminer. And this is now, the next two slides are a bit techy, talking about the technologies behind storing those networks. So Netminer knowledge graphs can have millions of relations and nodes between them. We've been working in the last few years, especially with Marco Brandisi, to make our knowledge graphs fair, findable, accessible, interoperable, and reusable. So we take lots of real-world data, and then we transform it into graphs, and Marco has developed tools to export those graphs into RDF and Neo4j and make it more interoperable so computers can easily access and we can connect Netminer more easily to it. So if you're more interested to really about this side of the work, then I just point you to some papers that we've recently published. They talk about how we go from our proprietary networks into RDF and what sort of endpoints can we develop that help computers easily understand the data? Okay, but for the purpose of this talk, I will go back and simplify things a bit again. So, right, so the problem we've got is we have built a huge knowledge graph with lots of different types of information. And the question now is, how can we interpret so much information and present it to the end users, to domain experts like you? So we need methods that can help us to evaluate millions of relationships in a knowledge graph and prioritize genes, extract relevant subgraphs, and we need tools that make the whole, all the subgraphs and all the data more interactively available to end users. So the next few slides will be about different use cases that we've been working on with some of you in the past years. And I have mostly focused on some recent small examples just to give you an idea how this knowledge graph can now be used. All right, first one. So the first one is a simple use case. So you've got a specific candidate gene and you want to know what does Netminer know about your gene. So think about it in a reverse genetics setting. So you've got a gene and you want to see what sort of traits is it linked to using the Netminer knowledge graph. Okay, so you go into Netminer and you enter your gene. In this case, it's TT2 into that list, and press the search button. And I will now explain a little bit what the story is that Netminer produces for this particular gene. Okay, so we are looking at the knowledge graph here, and the different symbols, they have a meaning. So TT2 itself that we inputted into the system is this blue triangle. And now by looking at the knowledge graph, we can see it's linked to things that we probably knew already. It's linked to pericarp color, seed coat color. But interestingly, it also has some links to seed dormancy and grain germination that are things maybe we were not really expecting. So the top bit of the graph is data coming from wheat, and then the bottom bit is where we enter through homology into Arabidopsis, and this is all the public information for TT2 in Arabidopsis. So the TT2 in Arabidopsis, we can see it's interacting with another gene called TTG1, which is then connected to root-related traits. Okay, so this was looking at so now this is the network, and when we study the network and we click on relationships, what we can do is we can extract actual text from the knowledge graph. So I just read you some sentences that we could be extracting from a knowledge graph about TT2. So TT2 on chromosomes 3D in wheat is predicted p-value 0.01, which is a property of the relationship, to regulate the transcription activation of MFT according to data from the analysis of 850 RNA-seq samples using GeneE3. Another sentence we could extract from the knowledge graph is, I'm jumping now to this bit here. So TT2 is interacting with MFT. MFT has been recently linked to grain germination. The knowledge graph has the paper and a sentence. Recent studies in both Arabidopsis and wheat have uncovered a new role of MFT and seed dormancy, in seed germination and in seed dormancy. So another sentence over here is extracted from literature and links MFT again to dormancy. Mapping analysis showed that MFT on chromosome 3A is co-localized with seed dormancy, quantitative trait locus, So the MFT also, like in Arabidopsis, another sentence from the knowledge graph, has a 3' UTR variant that has been associated with a specific p-value with increased germination rate after 56 days, and so on. So as you see, it's just those knowledge graphs are very content-rich. So studying the knowledge graphs by eye as an expert, you can they can lead to lots of different new lines of inquiries. So, for example, looking at this, we could ask the next question would be, could grain color and pre-harvest sprouting maybe be linked because of the TT2 gene is targeting MFD? Do white grain varieties have increased root hair density? Is there a link between root hair density and pre-harvest sprouting? So it just helps you to develop new hypotheses and new ideas. Okay, so that was a reverse genetics application. Now talking a bit about forward genetics applications, how we can go from phenotype to gene using the Netminer knowledge graph. Okay, so this is an example taken from the ARA-Pheno database. So we're looking at a trait called anthocyanin 22, and there has been a GWAS study that is linked to anthocyanin 22. Okay, is that working? Yes, so on the left-hand side, you'll see some information about the GWAS study, what the trait actually is described here, and then the p-values that, the significance p-values for which you will have significant genes. So how does this information look now in Netminer's knowledge graph? At the top, you've got anthocyanin 22 as a node. Again, it's linked to specific SNPs that were associated with that trait. The p-value is shown here. Then those SNPs are linked to a gene called FLS1, and the distance is shown as a property on the edge. And these are genes in Arabidopsis that are interacting with FLS1, DFR, CHS, F3H. So the knowledge graph is showing you that information, and you can now ask that question, say, what are the wheat genes that are linked to those 4 genes in Arabidopsis? And it shows you, it expands the knowledge graph and shows you that these are the wheat genes that are linked to the Arabidopsis orthologs, which are linked to anthocyanin 22. Okay, another example, and this is very recent work in collaboration with Guillaume Menard and colleagues. So they're interested in identifying direct seeded rice varieties that have improved seedling establishment, emergence, and vigor. What they use to go to candidate genes is they are exploring Erie's large germplasm of over 3,000 rice varieties. For a set of varieties, they phenotyped them using image analysis. They phenotyped it for early seedling traits, root, and so on. And the genotype and phenotype data go together then to do this sort of genome-wide association studies, where we find regions in the genome that correlate with a trait. So those regions can be very big. They can have tens or hundreds of genes. And what Guillaume and his colleagues were interested in to find candidate genes for all those different regions. So we've been working on a gene discovery pipeline that takes those regions and ranks the candidate genes within the region to suggest some interesting leads. So this is showing you Netminer, where a list of candidate genes in rice were entered into Netminer. They've been ranked, and users can now go and click on a specific candidate gene and actually see if the prediction that Netminer makes about that gene to be related to some of those traits makes sense. So again, so we've clicked on a specific gene in rice, which is this one here, and the knowledge graph is telling us that this gene in rice is part of 3 QTLs in rice. They are linked to traits like grain number per plant. And then on this side of the knowledge graph, we can see that the rice ortholog in Arabidopsis is linked to lots of different root-related traits. So, and these are phenotypes, for example, that are known about the rice ortholog in Arabidopsis. Okay, so all right. So the last few minutes, will be just about you know, what are our plans for the future? What is it we want to do next? Okay, so one thing we're always keen to do is to grow the knowledge graph, to go deeper and give it a bit more breadth as well. So there are lots of layers of information that we are not yet capturing, and we like to bring them in. So work in progress, this is areas we are currently working on. So Sandeep in my team sitting here is recently joined us. He's working on plants and crop aspects. His applications are about genetic diversity and multi-omics, how to use Netminer for that. Emma Bailey, PhD student in my lab, she's building an insect knowledge graph, and her applications are about integrated pest management. And then Dan Smith sitting here as well, he's working more on the pathogen side, integrating information about Fusarium and other pathogens with application on how can we use those knowledge graphs more for pangenomic data analysis. In terms of what sort of future developments we are planning to do with Netminer, so there's really three main areas. One is we like to make the search experience a bit more personalized search experience. The second area is better knowledge visualization, and the third area is working on more predictive network analysis. So for personalized search experience, what I mean is we like to add features where we can save and share your networks. You can like and dislike relationships between entities. This information is captured, and networks can be more personalized. So next time you search for something, it uses user data. Better knowledge visualization. We like to invest some time into finding ways to reduce the information overload that you get in those knowledge graphs using clustering approaches. And we like to build new ways of annotating information in the knowledge graph with your own user data, like gene expression data, overlay that onto the knowledge graph. And the predictive analysis is about how can we find research trends using publication and graph data. You know, what are the hot areas that people are working on? And how can we automate the story and hypothesis generation? So right now, users need to click on information in the knowledge graph to come up with interesting ideas. Can we automate that somehow? I'll give you just an example of what I mean with automating it. So you search Netminer for a specific gene, and what you get is this knowledge graph, and it takes a few minutes to click on things and understand what they mean. But the knowledge graph itself is well structured. There is a well-structured semantics of entities and relationships in the network. There are confidence values attached to the edges, and lots of literature information in the knowledge graphs. So we could work on ways of going from the knowledge graph back into human readable text. So we actually provide a message section or some interesting sentences for your papers without you going and clicking on things. So we can generate leads more automatically. Okay, so these are some areas we like to work on in the future. So last slide, so the Netminer impact. So Netminer had over 1,800 unique users last year, 68% of our users coming from non-UK countries. Netminer has been part of 3 grants recently submitted to BBSOC, one BBR with Kim, one as Lola with Freddie and with Matthew Paul. The Netminer code itself is open source. Version 3.0 was released in 2019. Developers are starting to contribute to our open source tools, which is great. And other bioinformatics resources like ReadIS, T3, and Ensemble are starting to link into the Netminer knowledge graphs. We are getting requests from other scientists to build Netminers for new species, like for sugarcane and honeybee recently. And we do get invited to run training courses and workshops. Been running them at EBI, PAC, and IB recently. So Netminer itself is also deployable as a private instance, and 2 companies, agri-food companies, have deployed Netminer on site, and several companies have shown expressed interest to build their own private Netminers. Okay, so I just really like to conclude with, not with a lengthy summary slide, but with something I saw in one of the last conferences I attended, and just give you some ideas. So something to think about: the right data in the right format, in the right hands, at the right time. In that case, it was a biomedical workshop, was saying saves lives. In our case, it's just showing that it's really important to have right data in the right format, in the right hands, at the right time. And it's a big challenge to do that, actually. So, okay, so now let's acknowledge the people I've worked with in the last few years. So a lot of this work wouldn't have been possible with Ajit Singh and Marco Brandesi and other members in the bioinformatics team that are contributing to building knowledge graphs and testing knowledge graphs. William Brown from IT is helping us with Netminer a lot. And then our collaborators who contribute either code to the Netminer code base or build their own knowledge graphs for other species. They're listed here. We've got people I like to thank that are more data contributors, like Cristobal Uai's group at JIC. They generate gene expression data. They don't have ways to visualize it. They collaborate with us to bring it into the knowledge graphs. Then really big thank you to the users who use Netminer and provide feedback, and which we do take serious and try to release features that are satisfying our users' wishes. And finally, the funding, our sponsors. So currently, most of us are funded by Designing Future Read, and some of us are funded through the Pest Genomics Initiative. So this is the acknowledgement slide that humans like you can read. You can read this and you can understand what it means. Okay. If a machine looks at this slide, it wouldn't really make much sense to a machine. So my question to you would be, after my talk now, how would we need to model this so it actually makes sense to a machine to understand that? It's a serious question. Okay, so it would need to look something like this. Okay, so this is the acknowledgement slide where things are not strings anymore, they are proper things. The relationship between them is explicit. So a machine can actually understand what different things mean. So we've got people there, we've got resources, we've got funding, they're all there, and it makes sense to a machine, and it makes sense to humans. Okay, thank you very much.